Okay. Rob, you're here. So, I'd like to introduce our plenary speaker, fresh off the plane and the train and whatever else. Rob Knight, who's the founding director of the Center for Microbiome Innovation and professor of pediatrics, bioengineering, computer science, and engineering at UCSD. He's published a number of books focusing on the impact of microbes, the immune system, co-founded the Earth Microbiome Project, the American Gut Project, is exceedingly well published, linking microbes to a range of health conditions, including obesity, inflammatory bowel disease, and more. And he's going to talk to us about a microbial dimension to human development and the potential for new approaches to human well-being. Do you need some water or anything? We'll get you some water.
All right.
Take your time.
That's okay. Normally I like to arrive early to set up the AV, so we're kind of keeping our fingers crossed on this one. And yeah, train is optimistic in the United States. I was in a taxi from the airport and the driver recommended I bail out for the last mile because I wasn't going to make it. So, great, thank you. Excellent, brought my own clicker. Having done this before. All right, great. Thank you. Well, thanks very much for the invitation to speak here, and sorry about the tight scheduling. My plane from Rome was delayed by an hour, which was kind of critical with how we'd set it up. So I'm going to tell you about a microbial dimension to human development and the potential it brings for new approaches to human well-being, both on the communicable and on the non-communicable diseases side. So the question I was tasked with tackling is how might a more holistic and complex view of the relationship between microbes and people change the way we approach health? And it's already changing how we think about ourselves as human beings. So I'd like you to take just a moment to think about what you saw when you looked in the mirror this morning as you got up and you were preparing to come to this conference. I saw an organism that's just 43% human, not just because I was in Europe and I hadn't had my coffee yet and was already jet lagged, but when we think of what makes up our body, each of us has 30 trillion human cells, but we have 39 trillion microbial cells. So by that measure, we're only 43% human, and our microbes outnumber us even in our own body. But you might think, well, it's the 21st century, who cares about cells anymore? Shouldn't we be thinking about genetics? And it's even more shocking when we think about that, because each of us has about 20,000 human genes, but astonishingly, and we just learned this through efforts like MetaHit and the Human Microbiome Project, the size of the microbial gene catalogue is more like 2 to 20 million microbial genes, encoding the vast majority of our unique biochemical functions and the vast majority of our antigens. And so by that measure, we're at best 1% human. And what's shocking about this is that we're ignoring 99% of our genes when we do GWAS or we do systems biology based just on the human genome. And the really terrible thing about this is that we're ignoring the ones that we can change, not the ones that are fixed when we're conceived, but rather the ones we can modify throughout our lifetimes to promote health. And most of these are microbes in the gut, so that's about 2 kilos of your biomass. But it's important to remember that the gut is not Vegas, and that what happens in the gut doesn't stay in the gut. And your microbes in the gut affect all kinds of things, from the obvious, like IBD, to the more surprising, like food allergies, to the even more surprising, like liver disease and cardiovascular disease, the molecular pathways for which have now been quite well worked out. And then shockingly, our microbes have a huge impact on our brain in diseases like Parkinson's, multiple sclerosis, autism, where again, we have detailed mechanistic accounts of how all this happens now. And this is just in the last 3 years for the most part. And what's shocking is we're losing these microbial genes right now. So when we study hunter-gatherers like the Hadza in Tanzania and other people living lifestyles that are much more characteristic of the last million years of human evolution rather than the last 100 years, what we find is entire major groups of bacteria, sorry, the pointer's not working there, entire major groups of bacteria that we find in pre-industrial people but we do not find them in anyone who's taken up farming, let alone anyone who's sitting in air-conditioned rooms like this one. And this includes entire phyla like the spirochetes, so huge branches of bacterial life we're losing right now. So it's kind of like we're taking what used to be an inner rainforest, beautiful with complexity and diversity, and turning it into a cityscape like the one we're in now, where only the rats and the pigeons survive. And this global microbial biodiversity crisis may have real consequences. So I'm sure you're all familiar with Rachel Carson's work from the 1960s, documenting how DDT and other pesticides wiped out the birds and other components of large-scale ecosystems. Marty Blazer, who was chair of medicine at NYU, not far from here, documented the same kind of thing for our inner ecosystems in his book, Missing Microbes, that documents how we're getting rid of microbes starting in early life and how this may be an important biodiversity crisis. And what's shocking is through the 20th century, as one disease after another, although some of these are making a comeback, regrettably, like measles, but as one disease after another caused by single pathogens from measles to TB fell to advances in medicine and public health, at the same time we see a skyrocketing of multiple sclerosis, of Crohn's disease, of asthma, of type 1 diabetes. And what's shocking is when this graph was published in the New England Journal, none of those chronic diseases were known to be linked to the microbiome, whereas today we know that all 4 of them and dozens of others are associated with the microbiome in humans, and we can cause or cure analogues of those diseases in animal models by manipulating the microbiome. So there's a very important question about whether our attempts to eradicate single pathogens have had unintended consequences on the rest of our microbiome. So to get a handle on the complexity of the microbiome, in the Human Microbiome Project, this was a large-scale NIH effort to characterize the healthy human microbiome. And you might have seen in Nature the results from the sequel, HMP2, looking at specific diseases, was just published last week. But in the HMP, we looked at about 250 healthy people, up to 18 sites in the body, collected 4.5 trillion bases of DNA, 1,500 human genome equivalents, which is a massive amount for 2012. And so the great thing is we had an unprecedented amount of DNA sequence data about the microbiome. But that was also the terrible thing about it. And to illustrate this, I'm showing you the first file of data from the HMP. This is the first 0.1% of the DNA sequence data in this file, and there's another 17,000 just like it. So we really had our work cut out for us, right? And this was a big problem. Actually, let me see if I can get my previews up here. Yeah, this is a big problem for medicine now because there's various companies, not all of which have been raided by the FBI yet, and also citizen science projects like the American Gut Project, where you can send in your stool sample and have your microbiome done. And so it's your doctor's nightmare that you're going to come into his or her office with a huge grin on your face and a list of like 1,000 species they found in your gut. And ask them, you know, hey doc, with all this data, surely you can tell me what's wrong with me, right? And what's your physician going to do? Refer you to colleagues in psychiatry for being crazy enough to think they can do something like that in the 12 beautiful minutes you have together? And so the goal of my lab is to make it not crazy and to let you compare yourself to other people and to yourself over time so you can make something out of those complex multivariate profiles. And when we take the HMP data and reprocess it with CHIME, the pipeline my lab's been developing for almost the last decade, we get this map. And for those of you who are aficionados of this stuff, what we're looking at is principal coordinates reduction of unifrac distances. What you really need to know is each point on this is a whole microbiome, but read out not by microscopy, but it's DNA. And 2 points are close together on the map if they have more similar microbes in terms of their evolution, and they're further apart on the map if they're more dissimilar, and an automated layout technique based on those distances. Now remember, these people are all healthy, so there's no disease on this map, but you could imagine all the things that might affect your microbiome, like whether you're old or young, male or female, thin or fat, maybe what time of day you collected it, maybe what you ate the day before. And when I color by the main factor, it's obvious what's going on. The different parts of the body are like different continents, with the mouth, the skin, the vaginal, and the fecal communities being completely distinct from each other. And to highlight this, I show you the mouth and the gut of the first person in the HMP. But we couldn't understand these distances in isolation. It wasn't until we did the Earth Microbiome Project, where we crowdsourced tens of thousands of samples from scientists around the world, that we could put a scale on this, because we could go out on the planet's surface and ask what 2 environmental microbiomes are just as different from each other as the mouth and gut of this one individual. And if you think of your mouth as kind of like a coral reef, complex mineralized structures, covered with biofilms that maybe your dentist bugs you about. The amazing thing is your mouth is as far from your gut in terms of its microbiome ecology as the water in that reef is from the dirt in this prairie. And we never expected that, right? A few feet along the length of your body can make as much of a difference to your microbes as thousands of miles across their surface in completely different environments. So this may lead you to wonder, how do we begin on these microbiome maps? Where do our complex microbial communities start from in the first place? And if you have dogs or kids as I do, you likely have dark suspicions about this, all of which are true, by the way. So I can match you to your dog fairly precisely by the microbes you share. And an interesting fact is that fatter people tend to also have fatter dogs and cats. So the obesity epidemic is not just in humans, and it may be transmissible microbially. But in all seriousness, where we begin, how we're born has a huge impact on where we begin and on babies' first microbes. So what I'm showing you here is work we did with Gloria Dominguez-Ballo now at Rutgers, where, oh, I see, so the pointer's not going to come up. And that one is too, oh, there we go. So if you come out the regular way, that's in pink there, all of your microbial habitats all over your body look like your mother's vaginal community in red. Whereas if you're delivered by C-section, all of your habitats, shown there in light blue, including the gut, look like the skin community instead, shown in dark blue. So you start off in a totally different place. And Gloria and Marty have shown that antibiotics, birth mode, and diet interact to shape the microbiome in early life. And we've confirmed this with Merete Expo at the Norwegian Public Health Institute, where we can predict a lot about the baby's, the child's weight and cognitive performance at age 10 from their microbiome a few days after they're born. But there's hope because breast milk in particular seems to reverse the adverse effects of the combination of antibiotics and C-section, which has a large impact both on microbiome and phenotype. And at UC San Diego, we have a large newly opened breast milk research center led by Lars Bodie, where we're trying to understand what components of breast milk could be added to formula for families that can't breastfeed, as for example did not work for my own family, to be able to confer those benefits for everybody, not just those who are able to breastfeed. So that's where we begin. You might wonder where you go from there, and I'm sure you've all seen children develop physically, mentally. Now you're going to see one develop in terms of his microbiome. And these are all fecal microbes, and we're going to connect the frames of this animation one week to the next as he moves through that microbiome map. And you can see that yellow dot. These are fecal microbes, but he starts off in the vaginal microbiome part of the space, as we would expect from his delivery mode. And so he's going to develop towards the fecal adult state defined by the Human Microbiome Project at the bottom there. And so the question is it going to be fast or slow, complete or incomplete over 2.5 years? Does anything happen along the way? And I start this going, you see some weeks there's tremendous change one week to the next, others there's just a little change. And the main thing that matters on these maps is distance. So the kid is more different from himself one week to the next than the difference between any two healthy adults in the HMP. So if you think your child's a different person one week to the next, it can literally be true in terms of his microbiome, where most of the genes are in the first place. And coming up here is something that's absolutely shocking. So he gets antibiotics for an ear infection. You see this massive regression of the microbiome, followed by rapid recovery. And that went by pretty fast, so I'll just rewind it for you and play it for you again. What we see is on dose of oral amoxicillin for ear infection, we see this tremendous unraveling of the microbiome, unwinding months of normal development, but then he bounces back in just a couple of weeks. And by the time he reaches 2.5 years, he's essentially in the healthy adult state. In case you're wondering, that's when he was toilet trained, and it's a lot easier to get the fecal samples out of a diaper than to fight a toddler who's probably learned how to flush for every specimen. So a lot of studies tend to end at about this time. But although this child was resilient to antibiotics in terms of his microbiome and phenotype, a lot of children and a lot of adults aren't. And so one major challenge in the field is to find out why and if we can predict who's at risk. And these development effects are so strong you can pick them up cross-sectionally. So this is work we did with Gloria, with Jeff Gordon at WashU and a number of others, where what you can see is approach to the adult state of the microbiome over the first 3 years of life in an African population in red, a South American population in green, and the US population in blue. And in all cases, the approach to the adult state is pretty much complete by age 3, and then it stays in the same region. But where they wind up is completely different. So here's the principal coordinates map of the same data. And you see the main driving effect is age from the youngest kids to the oldest adults, but the final state is totally different between the Western and the two non-Western populations that have totally different disease profiles for both infectious and non-communicable disease. And in this respect, it's important to remember that the HMP and MetaHit and most other large-scale projects are still focusing on Western adults. And as soon as you look at kids, and as soon as you look at non-Western populations, you see completely different configurations of the microbiome. So one thing we need to do globally is to build microbial growth curves for children for diverse populations. We're starting at Rady Children's Hospital in San Diego, but we need to do this everywhere where the microbiome backgrounds are different to measure the impacts of breastfeeding, of antibiotics, of delivery mode, of maternal drugs, and see when babies are falling off this microbiome growth curve and what impact it's likely to have. So then you might wonder what sorts of research should be encouraged to help us have a more realistic understanding of this complex relationship of microbes to infectious diseases, to non-communicable diseases, and to normal physiological functioning. And I'm sure you've heard a lot already about how our war against antimicrobially resistant microbes is failing, and new approaches are urgently needed. And a lot of work has gone in combating individual species or whole taxonomic groups of microbes. Much less has gone to strengthening the host, which I think has a tremendous amount of potential. And it's well known that one way to strengthen the host is through diet, but it turns out that the details of how you strengthen the host against infection and against chronic disease through diet are relatively challenging. So for example, it turns out that you can't make that shield out of iron. What I mean by that is that there have been many attempts to supplement with iron anemic populations in childhood, and in general those attempts release pathogens that are limited by iron, and so they're able to take off and cause diarrheal diseases and other problems. And if you look at an example like Salmonella typhimurium, what actually works is a combination of copper, of arginine, of vitamin C, and of linalool. But each of those components was worked out painstakingly, one step at a time, in individual studies. And what we need is a more global approach to much more rapidly profile for a huge number of pathogens, for a huge number of micronutrients and dietary ingredients, what will have an impact. And this is not just bacteria, by the way, but also viruses, at least in animal models. And this paper from Tad Steffenbach's lab, it's in a mouse model, but it was absolutely astonishing. So what he found was that normally about half of C57 black 6 mice die when they're exposed to influenza, but you can reduce that to just 10% mortality by supplementing with a molecule called amino tyrosine, which is naturally produced by gut bacteria from flavonoids in the diet. And so if you give them antibiotics, essentially all of them die, but you can rescue a lot of them with DAT. What's more, you have to have the right microbes, so you have to have Clostridium orbicindens, not other Clostridia or Enterococci, but you need something that's actually going to make that compound from the dietary ingredient. This is why we have to integrate diet and microbiology and immunity together to get a full picture of this stuff. So to start addressing this problem, Peter Darrestein and I recently launched what we're calling the Global Foodomics Project, where what we're doing is we're taking thousands of food samples and running them through the mass spec to find out what's really in them at the point of sale or at the point of consumption. And then we're cross-referencing the data with the American Gut Project, where we have over 20,000 stool samples that have been sent in by citizen scientists, and also with our various clinical projects to do meta-analyses to relate what goes in to what comes out. And this is a picture of the molecular network from an early iteration of this project. So what you need to know is that the nodes are different chemicals. There's an edge drawn between them if their spectra from mass spec are more than a similarity threshold, and they're colored by source. So the green ones are from food, the brown ones are from what comes out the other end, and the blue ones were found in both sources. And you can see that there's not much blue on that diagram, which speaks to the complete biotransformation in our gut of the things that we eat. So to find out more about that transformation, we've been turning again to mouse models. And so with Sarkis Masmanian at Caltech, we've been scanning mice in 3D and then dicing them up into little pieces and then sequencing and running through the mass spec each piece of the mouse. And so what we can do is we can reconstruct a 3D model where we can highlight any bacterium or any molecule in the full 3D structure of the mouse. And we're doing this for conventionally raised mice that are called SPF and what you're going to see, specific pathogen-free. And we're also doing it in completely germ-free mice. So here's just one example of the sort of thing that we see. So if you take a molecule like soya saponin, the scale on this is from blue as the least to red as the most, and you can see it builds up in the germ-free mouse's gut, but you don't see much of it in the SPF mouse. Whereas in contrast, soya sapogenol is created by cleaving this bond, And you see it accumulates in the SPF mice that has the bacteria that do the reaction, but it doesn't accumulate in the germ-free mice. And both soya saponin and soya sapogenol have been shown to modify immune response. And we also see a complete transformation of the bile acid profiles and many other details of what's going on in the mouse's metabolism when we do this kind of work. So you might wonder, does all this diversity in the microbiome really matter? And I told you about a number of diseases that have been linked to the gut microbiome, including things like inflammatory bowel disease, irritable bowel syndrome, that might seem obvious, things that are more surprising like food allergies, like asthma, like cardiovascular disease, like diabetes, and then things that are really surprising like multiple sclerosis, autism, and Parkinson's. And one thing I've worked on for about the last 15 years is obesity with Jeff Gordon at WashU and a number of other collaborators. And today, for example, I can tell you with about 90% accuracy if you're lean or obese based solely on sequencing the microbes in your gut. So on the one hand, this is a cool trick from a technical perspective. On the other hand, we don't think it has a lot of medical or commercial potential because I bet you can tell which of these people is fat without doing any sequencing at all, right? But an enormous amount of money has been poured into looking at the human genetics of obesity. And if you take every SNP ever linked to obesity by genome-wide association studies, you can do that classification task, lean or obese, with about 57% accuracy from human genes versus 90% accuracy from microbial ones. And it's not just our lab. So Erin Segal's lab at the Weizmann Institute has shown that for a large number of traits, what you're seeing here is the microbial contribution in green and the host contribution in blue, where for a lot of things like lactase consumption, for waist circumference and so on, the microbes dominate, whereas for other things like height, it's pretty much all host genetics. And what's amazing about this is if you want to associate a trait with a microbe rather than with a human gene, you can typically do it with 10-fold fewer subjects because the associations are so much stronger. Although you have to be careful, we've shown with Hongwei Zhao at Southern Medical University in China that if you train a model for healthy versus disease in one city, and then you try to deploy it in another city, even in the same region, the same microbes that make the difference between sick and healthy in one place also make the difference between healthy in one place and healthy in another. So you have to retrain the models right now for every population you want to look at. This is why a coordinated global effort is needed. And what I've been telling you about so far has mostly been based on association, but what about causality? Well, to assess causality, we moved to this technology called notobiotic mice. So this is Jeff set up at WashU, where what we're doing is raising mice in a bubble with absolutely no microbes of their own, and then we colonize them with microbes that we think will make an impact on their phenotype so we can establish causality. And Vanessa Radara and Jeff's lab infected these germ-free mice raised in a bubble with microbes from an obese human, and what she got was obese mice. Whereas in contrast, if you infect them with microbes from a lean human, the mice stay lean. And so this has been done not just for obesity, for IBD, for colon cancer. Sarkis Nasmani and I have done this for Parkinson's disease, for multiple sclerosis, and most recently for autism, which came out in Cell just a couple of weeks ago. But also all kinds of other traits, including response to drugs, ranging from things like acetaminophen to the latest checkpoint inhibitors for cancer, response to diet, including things like emulsifiers and artificial sweeteners, where your response is highly individualized, and then the susceptibility to a wide range of bacterial, viral, and fungal pathogens can be transmitted from humans to mice on an individual level by this sort of approach, establishing that those microbial genes can be causally implicated, at least to the extent that the animal models are a good model of these diseases, which for things like autism, you can argue about. So in summary, what we need is we need more work that integrates food, drugs, microbiology, and immunology together into one study. And the drawback is that this stuff is expensive, so it's often problematic to get one study that will fund all these data layers with enough people to get adequate statistical power. We need more comparative studies of microbiomes in populations where the burdens of infectious and chronic diseases differ. And the microbiome is totally different in different places around the world, but especially places where, say, IBD is increasing rapidly, like Asia, would be especially important to target with a view to intervention. We need more prospective longitudinal studies that integrate microbiology and immunology and infection and chronic disease. So frequently, longitudinal studies only look at one of these techniques and only look at one of these readouts, but we really need to do piggybacking, for example, on studies to add additional assays. We need more translational work so we can understand what features of animal studies are going to translate back to humans and what is not. We need more longitudinal studies that are not just observational, but have defined perturbations, especially using already approved compounds and supplements and dietary ingredients, so there's not a long FDA approval process, and especially in early life, where, as I showed you, the microbiome changes the most, and where a lot of permanent things happen during those sensitive and critical periods for development. And then we also need better molecular tools to read out everything faster, cheaper, with more detail, and better data science tools to make sense of what it means. So I'm going to show you the best example we have of why you care where you are on those microbial maps, like I just showed you in the HMP. And this is just to reorient you on the map. And I'm going to show you some work we did with Alex Karutz and Mike Sadowski at the University of Minnesota on patients with C. diff, which is a very important hospital-acquired infection that's increasing in incidence rapidly in the US. And these spiky shapes, these are stool samples from people with C. diff, and you see they look absolutely nothing like healthy stool. And remember, the distance from here to here is the distance between dirt and seawater, right? So we're talking about a profoundly altered microbiome. What's going to happen is 4 of those patients are going to get a fecal transplant from one donor. And if you're wondering, and you can see the donors in the healthy region defined by the HMP at the bottom. And if you're wondering what a fecal transplant is, here's Bill Sanborn, our chair of GI, about to deliver one using hospital-grade stool he got from a nonprofit called Biome, because in the US, the FDA regulates stool as a drug. It's pretty difficult to prove you use GMP and GLP, right? But somehow you have to put in the effort. So anyway, 4 of those patients are going to get a fecal transplant from that one healthy donor. But unlike the infant time series I showed you, here it's just going to be, each frame is just 1 day in the lives of these 4 patients. And you might wonder, is a few grams of stool even going to change their microbiome ecology at all on the scale? And what you can see is that in just 2 or 3 days, they completely transform from the unhealthy state up here into the healthy state. And all of their symptoms are gone in those 3 days. So they're producing firm stool for the first time in years. They had to fail antibiotic therapy for at least 2 years to be eligible for this trial. And so what's remarkable about this is that each of those time points, you're talking about thousands of microbial taxa, and yet we're turning this into an axis. That basically goes from sick to healthy along one dimension. That's much easier to explain than those profiles and changes in those profiles are, and that's easy to visualize. And so what we need to do now is for all these other disorders that have now been linked to the microbiome by association studies in humans, and also where we have causal evidence in animal models that the microbiome can cause or cure them in those models, we need to find the good and the bad places on the microbiome map across different populations, across different medical specialties. But we also need to go beyond that and develop a kind of microbiome GPS that tells you not just where am I right now and where do I want to go, but how do I get there by changing the microbiome, whether it's by something that seems as extreme as fecal transplant or phage therapy, which we're also doing at UC San Diego, whether it's something in the middle like drugs or probiotics or prebiotics, or whether it's reshaping your microbiome through diet, which in the American Gut Project we've shown is one of the most powerful ways you can reshape your microbiome, but it takes 6 to 12 months. So what you ate at lunch doesn't matter at all. What you eat for lunch for the next 6 months is going to matter a lot. So we need to develop this kind of microbiome GPS and make it so easy to use that even our children could use it. So you could imagine a smart toilet where as soon as you flush, it delivers the results of your data dump, as it were, to your smartphone, which I bet you're using in there anyway, and tells you, am I going in a good place or a bad place? And when my daughter was 2, when we took this photo, she'd already learned how to use a smartphone and regrettably where we often use them, but we couldn't do this kind of data display on a phone, whereas now we can, and this is her own data over the first 2.5 years of her life going towards the adult stool, oral, and skin states. And this is the kind of thing that you can explain to a 7-year-old on an iPad, given enough time. And then the question is, can we make it easy enough to explain to a patient in the context of a very short clinical visit? But ultimately, this stuff still relies on million-dollar sequencing equipment and mass spec and so on. And what we want to do, and this is really the science fiction part of the talk, we want to get it into everyday use, where the vision is that as you examine yourself in that mirror in the morning, as you breathe in the mirror, you could do some sort of instant chemical analysis on your breath by mass spec or NMR or Raman spectroscopy. Obviously, we're still working on the user interface for this part because you probably don't want to see that first thing in the morning. But then the idea is to use some of the same deep learning technology that backs Google Translate to translate that into a microbiome profile like you would get from American Gut, and ultimately place you on that microbiome map, where if your location on that map puts you at risk for anything, it'll tell you, and then either tell you to consult a physician or tell you about things that you could do yourself that would modify your risk based on the best evidence that we have available. And while we're talking science fiction, you could imagine your mirror talking back to your smartphone over the Internet of Things, so that later, when you're faced with 1,000 yogurts in the supermarket, you can use augmented reality to zoom in on the one that you're looking for, and then pick it off the shelf and scan the barcode to confirm that you got it. And then you could also imagine your smartphone recording things you did during the day that affect your microbiome, like what you ate, who you were with. Like, we're all exchanging microbes right now just by being in this room together, and you can measure that after a few hours. How many minutes you spent outside, which has an impact on your gut microbiome, how far you were from open space or from a beach, all these different things, and integrate, and then take that and integrate it and feed it back to your mirror, so your mirror can edit your video image in real time and show you visions of yourself 5, 10, 20 years in the future if you behave every day the way you did today, or what you might look like if you did a little better or if you did a little worse. So you laugh, but most of the pieces of that exist already, including the live video editing, not just for weight and for age, but also for things like affect. And so the kind of thing we're doing at the Center for Microbiome Innovation is bringing together over 140 faculty members and a growing list of corporate sponsors, because this is the kind of thing we're not just going to be able to do in a university setting, but if we want to manufacture it and distribute it, we're going to need to get it out there into the world with corporate partners. And we're also doing a number of other things as ambitious as that in other areas like the environment, industrial settings, and so on. So my time is up. I'd like to just briefly thank over 1,000 collaborators, some of whom I've named, but I'm not going to read out the whole list. The people in my lab, who it's an amazing privilege to be able to work with, our various sources of funding, including the tens of thousands of members of the public who've contributed to American Gut. And finally, I'd be delighted to take any questions. Thanks again for the invitation to be here, and I'm looking forward to the discussion. Thank you.